So this week we're going to focus on just chapter 14, and this is going to cover the basics of inheritance, so how genes are passed on from one to the next, and we're going to use Punnett squares to look at how genes are inherited. The principles of genetic inheritance were discovered by a monk named Gregor Mendel, and he did this in a garden with pea plants where he looked at specific traits and just looked at many plants over many generations. And when you think about kind of the tools that he had available to him, it's amazing with the principles that he was able to deduce from just breeding plants for many generations. So here are some terms that we need to clarify. A character is some kind of heritable feature. In the plants, Mendel looked at several different characters. Character could be the flower color. Is the plant tall? A character can have many different variants. So each variant is called a trait. So if the character is flower color, there are several different traits. So it could be a purple flower or a white flower. So Mendel was able to track the occurrence of these specific traits over many different generations. What he did was he chose characters that had only two distinct forms, so tall, short, purple, or white. Mendel started with plants that are called true breeding. A plant has a specific trait, and they pollinate with themselves. Remember, plants can reproduce with themselves, that they're always going to produce that same specific trait. So that trait breeds true. For example, a white flower plant breeds with a white flower plant, and all of the offspring are white flower plants. So that plant would be considered true breeding for white flowers. So here's Mendel's experiment, and we'll just use one example of flower color, but he did this for 8 different characters. In this case, we have a true breeding purple flower plant and a true breeding white flower plant. So both of those, if they mated with themselves, would produce the same color. So Mendel wanted to see what happened when I take these 2 true breeding varieties and mate them together. This is called the P generation for the parent generation. The P generation is always going to be true breeding parents. So what happened when he did this was he found that the next generation, this is called F1 for filial 1, meaning this is the first generation after the parents. These were all purple. So for some reason, the purple completely covered up the white, and there were no white flowers in the second generation. We know now that these individuals were hybrids. At the time, Mendel didn't know that. We know that they have one allele for purple and one allele for white, and that the purple was dominant, and so it covered up the white allele. Mendel didn't know this. So Mendel had this question at this point: is the white gone? Did purple completely erase it? If I mate this plant again, will I see white? What happened to the white? The next generation is called the F2 for filial 2, where he mated this with itself. So this is self or cross-pollination between all of these individuals. So it's mating either with itself or with its siblings. And what he found was that most of the third generation here was purple. So this is about 3/4 purple plants. But the white reappeared. We had about 1/4 white plants. So now Mendel knew that the white didn't get lost. However, for some reason in the second generation, that F1 generation, the white flowers got completely covered up. And then they reappeared in the third generation, the F2 generation. So what he deduced from this is that the purple flower color was dominant, meaning that it dominated over the white. So when they were both available, purple was chosen. And the white flower color is a recessive trait, meaning that it is completely covered up by the purple when both of them are present. What he found by doing the F2 generation was that it was not diluted or destroyed. The white flowers came back as fully white flowers. So it was either purple or white. There was no mixing or blending or dilution of these traits. So Mendel decided that there must be some heritable factor that delivered the trait either in its entirety or not at all. And so what he was considering a heritable factor is now what we call a gene. So this gene is a sequence of DNA, and now we know that the gene would either code for purple flowers or white flowers. And if they had the gene for both, if they had one gene for purple and one gene for white, because purple was dominant, it would completely cover up the white, and you would get purple flowers. So now we're going to go through the many contributions Mendel made. So these are the conclusions that Mendel came to as a result of his experiments. So the first conclusion was that alternate versions of genes account for the variations in inherited characters. This means that that heritable factor, the genetic factor, is what causes the different traits, whether it's purple or white. So we call these different versions of the genes alleles, and we usually represent them with a letter. The letter is usually going to correspond to the dominant gene. So if we're talking about purple versus white flowers, usually we'll use a capital letter P to indicate the dominant allele. And we use P because the dominant was purple. And then we keep the letter the same to make sure that we know that we're talking about the same character. And we might use a lowercase p to talk about the recessive allele. If an individual has a dominant P, this is a trait that codes for purple, or the dominant trait. And then an individual with a lowercase p will code for the recessive trait, the white flowers. He also decided that these genes have a specific place on the chromosome. So if this is the chromosome, the gene will always be in a specific location or locus. So a locus is just a specific location on a specific chromosome. The second discovery that Mendel made was that for every character, so for example, flower color, an organism gets two alleles. We get two letters or two versions of the gene, one from each parent. So in the case of flower color, each plant gets one allele for flower color from its mother, and it gets one allele for flower color from its father. And then the combination of those two alleles will determine what flower color that plant actually has. So sticking with our flower color example, there are three options. You can be capital P, meaning that you got the purple color allele from both parents. Or you can be little p, meaning that you got the white allele from both parents. These are both considered homozygotes. This word homo here means same. So in this case, these both have the same letter casing. So big P, those are both capital. Little p, those are both lowercase. So this individual would be called homozygous dominant. And this one would be homozygous recessive. There's one more option, and the other option is that you can have both, a big P and a little p. These organisms are what we call the hybrids. So these are going to have two different alleles. So these are called heterozygotes. Hetero here means different. Another term for these is hybrid. So that entire F1 generation were heterozygous. So these are heterozygous. The homozygous individuals can also be called true breeding. So we know that all of the true breeding parents in the first generation were all homozygous, and that's why they produced all flowers of the same color. And then all of the individuals in the F1 generations were hybrids because they had one big P from one parent and one little p from another parent. These are what we call genotypes. This is the actual genetic sequences that each individual carries in its chromosomes. The third conclusion that Mendel came to is that when the two alleles at a locus differ, so specifically talking about heterozygous, the dominant allele, big P, determines the organism's appearance, and the other allele, the recessive, the little p, has no noticeable effect. So this is called complete dominance. So this is a heterozygous individual. We can use the genotypes to figure out the physical expression of these genes. What color are the flowers? The phenotype is the physical expression, whereas the genotype is the alleles. So an individual that has big P, those are both dominant for purple flowers. The phenotype is the actual trait. This plant is going to have a purple flowers. So that's the physical expression. The other homozygous little p, those are both recessive. So those are both going to code for white flowers. So it makes sense that this plant would end up with white flowers. So what this third conclusion says is that when an individual is big P, little p, meaning that it has one capital and one lowercase, one dominant, one recessive, it's actually going to have purple flowers. It's not going to have any white. It's not going to be like diluted or mixed. The dominant purple trait completely covers up the recessive white trait. And so often when we talk about, this is called complete dominance, or sometimes we equate this to the term called Mendelian Genetics. The fourth main conclusion that Mendel made is actually now got its own name. So this is called the law of segregation. This is an important one for understanding how Punnett squares actually work. So what it says is that the alleles for a heritable character separate or segregate during gamete formation and end up in different gametes. So this is happening during meiosis. So here I have a diploid cell, and this individual, I'm going to use big P, little p, just to say that's the genotype of this individual. Diploid means that I have both sets of chromosomes, right? So they have one chromosome here, another chromosome here on the homologous chromosomes. Separation of homologous chromosomes during meiosis. So we're going to do meiosis, and we're going to end up with gametes. This individual can pass on eggs or sperm that have half of the number of chromosomes. So right now we're just focusing on these two chromosomes right here. The two options for the gametes, let's say this is an egg. The egg could be big P, or we can make eggs with little p. You're not going to make eggs with both of them in there. So this is the idea that during meiosis, these two are going to separate from each other, and they're going to be inherited independently, or they're going to separate. And so we end up with gametes with only one allele for each character. So this individual can pass on a dominant allele or a recessive allele, but not both. We've already seen that the genotype is the actual alleles, the genetic makeup, and the phenotype is the physical appearance. And we can always go from genotype to phenotype. So given the genotype of an organism, it's easy to decide what the phenotype is going to be because we know the genetics. You cannot always go from phenotype to genotype. It's hard to distinguish what the genotype of a plant is from just the phenotype. So if the phenotype in this example is white, we can figure out the genotype. When we're looking at the recessive trait, you are going to be able to figure out the genotype. You know that it has to be little p, little p. That's the only way to get white flowers. However, this is an example. This is the F1 or the F2 generation here, where we got 3 purples to every 1 white. We have 3 purple, which is the dominant trait. These actually have 2 different possible genotypes. They can be big P, or big P, little p. You cannot look at a purple flower and determine its genotype from just the color of a dominant trait. So when you have a recessive trait, yes, you can figure out the genotype because there's only one possibility. However, when you look at a dominant phenotype, like purple, you have 2 different options for what its genotype could be. So we use a tool called a Punnett square for determining the ratio or the genetic possibilities of offspring in a cross. So let's look at Mendel's experiment and show you the Punnett squares for the different generations. In the P generation, Mendel started out with 2 true breeding plants. And we know that one of them was true breeding for purple flowers, so that is big P. And they mated that with another individual that is true breeding for white flowers, so little p, little p. And I like to just draw the little tail on there to help me distinguish these letters from each other. Next, we know the law of segregation. Each of these plants can only pass on one allele at a time. So this parent can pass on, let's say this is mom, an egg with a big P or an egg with a big P. This parent, let's say it's dad, can pass on a sperm with a little p or a sperm with a little p. Those are the same because they're true breeding. So what we do is we're going to line up a Punnett square. So a Punnett square is going to take the gametes, not the parent genotype, the gametes, and arrange them in all possible combinations. So I have 2 gametes from mom, 2 gametes from dad, so I need a 2 by 2 Punnett square. So I will take the mom's gametes and put them over here, and dad's gametes and put them over here. Then all we have to do is combine all of the possible combinations, and we just drag them across and drag them down. So big P, little p, big P from mom, little p from dad, big P from mom, little p from dad, big P from mom, little p from dad. And just a convention, you always draw the capital letter first and then the lowercase letter. It doesn't actually matter what order they're in, but to make it simpler, just do capitals first and then lowercase. So now we can see that these individuals make up the F1 generation. So if you remember what Mendel saw, is he saw that the F1 generation was 100% purple flowers. And so here we can see that represented in the genotypes. We've got all 4, so 4 out of 4 are big P, little p. So they're all heterozygous or hybrid. And in here we can see the big P completely covers up the little p, and we get all purple. So then he did another cross. Mendel mated these individuals with each other. So next we have the F1 cross, where he mated an F1 individual, which is big P, little p, with their sibling, big P, little p. Remember, this is plants, so that's okay. So again, we have the law of segregation. This parent, let's say mom, can make an egg with a big P or an egg with a little p. And then this parent is dad, can make a sperm with a big P or a sperm with a little p. And then each one of them will pass on one allele. So we have 2 possible alleles, or 2 possible gametes from mom, 2 possible gametes from dad. So we need a 2 by 2 box. So let's put mom over here on the side where we've got big P or little p. Dad we put over here on the top, and it doesn't matter which one goes on which side or the top. And then again, we're going to drag them over and down. So in this square, we will get big P, big P from mom, big P from dad. In this square, again, we get big P from mom, little p from dad. In this one, we get big P from dad, little p from mom. It doesn't matter where they came from, just capitals first, then lowercase. And then in square, we get little p from mom, little p from dad. So this box here represents the F2 generation, where Mendel saw we had 3 purple to every 1 white flower. And so what we can see is the big P, this is going to be a purple flower. Big P, another purple flower. Big P, another purple flower. So we get 3 out of 4 are purple, with either big P or big P, little p. And then we have this last one here, little p is the 1 out of 4 that is white with little p. Oftentimes we summarize these Punnett squares with the genotype ratio. And so a genotype, again, is looking at the alleles, where we have 1 big P for every 2 big P, little p's, for every 1 little p. So this is one way to kind of describe what's happening here. And then the other description we often use is the phenotype ratio, which again, phenotype is the color, where we would have 3 purple for every 1 white. So you should expect to be able to get questions where you have to draw a Punnett square and then describe the ratio of the the offspring, either their genotypes or their phenotypes, like this. What we saw in the F1 offspring there is that the cross was a monohybrid cross. Mono means that we're looking at one character. So we're only using one set of alleles for each parent. And hybrid, of course, means that they're heterozygous. So when we had big P, little p, crossed with big P, little p, this is one character. We're just looking at flower color. And both parents are hybrids, where they have one big, one little, one big, one little. And so a monohybrid cross is always going to give you a 3-to-1 phenotype ratio and a 1-to-2-to-1 genotype. So let's go back to the question of, I hand you a purple flower. And the question I have to ask you is, what is the genotype? Well, you don't know. From just handing you a purple flower, it could be either big P, or it could be big P, little p. Both of those will give you the exact same purple flower. So in order to figure out what the genotypes were of his F2 cross, because Mendel didn't know whether the purple flowers were big P or big P, little p, he did a test cross. So a test cross is when you're going to take your purple flower and you're going to mate it with a homozygous recessive individual. So I'm going to mate this with a true breeding white or little p individual. So let's go through an example of a test cross. There are two options. I handed you a purple flower. It is either big P, or it's big P, little p. One of those two is true. So what I'm going to do is I'm going to mate it with a white flower. So I'm going to draw two crosses here to show you the two options. And then based on what you would physically see, you would be able to decide which parent you had. This individual can make alleles or gametes of big P. This individual can create only little p or little p, right, sperm. We'll go ahead and draw our 2x2 Punnett square. So big P, little p, and then fill in the square. And this should look just like the P generation cross from Mendel. And the result of this is we will get 100% purple flowers. So if you did this test cross and got 1,000 individuals, you would expect all of them to be purple. The other option for your plant was that it was big P, little p, in which case mom here could make a big P or a little p, and dad could make a little p or a little p. So then you can draw the Punnett square, 2x2. Here's mom, big P, little p. Here's dad's gametes, little p. So then we combine them, and you can see that they make 2 heterozygous and 2 homozygous recessive. So from this, we would expect 50% to be purple and hybrid, big P, little p's, and 50% to be white, little p. So if you did this cross and got 1,000 offspring, you would expect about half of them to be purple and about half of them to be white. So what you would do is you would then observe the offspring of this cross, and if you saw any white flowers, and it should be about half, you would have to conclude that your parent was big P, little p. And if you saw no white flowers, you would conclude that your parent was big P. The last law that Mendel described was the law of independent assortment. And this says that every pair of alleles segregates independently of any other pair of alleles. So now in this case, we're going to look at 2 different characters. We'll keep our flower color, which is P, and then we're going to look at another character, let's say maybe plant height. So in plant height, a tall flower is dominant, and a short flower, we'll use T again, would be recessive. What Mendel's law here says is that the alleles for plant flower color, P, and the alleles for plant height, T, they don't depend on each other at all. P will assort by itself randomly, and T will assort by itself randomly. So they're not linked to each other, they don't depend on each other at all. The way that Mendel showed this is he used what's called a dihybrid cross. Di means 2. We're going to be looking at 2 characters. We've got flower color and plant height. And hybrid means that both parents are hybrids for both traits. So the parents are going to be big P, little p, big T, little t. That is a dihybrid individual. They have two traits, P and T, and they are both hybrid, heterozygous. So let's look at an example of a dihybrid cross. So we are going to take two dihybrid individuals, big P, big T, little t, and cross them, big P, big T, little t. The first step is always figuring out what are going to be the gametes. Before this was pretty simple. You just took the two letters and separated them. Now we have to make all possible combination of gametes. Remember that a gamete will have exactly half of the traits. But you don't want a gamete to have two P's or two T's. Each gamete needs to have a P and a T, because they're going to pass on one allele for each of these two characters. We can make all of the different possible combinations. The trick I like to use, if you've been in algebra before, is called FOIL, to figure out the 4 possible gamete combinations. These both parents have the same genotype, so they're going to have the same gametes. So I'm just going to draw this here, big P, T, little t. So F stands for first. So that is going to be the first of each character. So the first is going to be big P and big T. So big P, big T. O stands for outside. So those are the two on the outside. So the outside is going to be big P and little t. So that's going to be big P, little t. And each of these is a gamete. Like those might be an egg. I stands for inside. So those are the two on the inside, the little p and the big T. So the next egg would be little p, big T. And then L stands for last. So that's the last one in each pair. So little p, little t. So with 4 different alleles here, there are 4 possible combinations of gametes. So these are the gametes. Each parent is going to be able to make the same 4 gametes because they have the same genotype. So if this is mom, she can make 4 eggs. Big P, big T, big P, little t, big T, or little p, little t. Dad can make the same 4 sperm. Big P, big T, big P, little p, big T, little p, little t. So now I'm looking at the number of gametes, and I have 4 from mom and 4 from dad, and that's more than a 2x2 square. I'm going to need a 4x4 square for my Punnett square. So then we do the same thing as a normal Punnett square, and we're going to take the gametes and put them on the sides. So I'll put mom on the side here, big P, big T. They go together. Remember, this is one gamete. Big P, little p, big T, little p, little t. And then dad goes on the top, big P, big T, big P, little p, big T, little p, little t. Each gamete gets its own row and its own column. You can do a quick check to make sure that you're doing this right. Each of these parents has 4 genes, or 4 chromosomes. This is the 2N diploid status. And each of these gametes here has 2 chromosomes, 1P and 1T, which is the N haploid gamete. And then when we combine them back into an offspring, each of these box represents an offspring. This is going to be a zygote. That's a diploid cell. So you combine the same way across and down. This individual should have 4 letters. Big P from mom, big P from dad, big T from mom, big T from dad. The convention is to put like letters together and then always capital before lowercase. So we put the P's together and the T's together. That's going to make it much easier for you to read what they are. So now you can check your Punnett square against mine to see if it matches. You should get the exact same Punnett square. When we describe these offspring, there are many different genotypes. We don't usually talk about the genotype ratio of a dihybrid cross. However, there are 4 possible phenotypes. A plant can be purple, and tall. Those are the two dominant traits. A plant can be purple and short, so purple being dominant, short being recessive. A plant can be white and tall, or a plant can be white and short. So when we look through this Punnett square, we have a total of 16 boxes, which means that if we count each of these different characters, we should come up with 16 as the total number. So let's start with finding all of the plants that are purple and tall. This means that they have a capital P and a capital T. They can be homozygous dominant for both, like this one here, big P, big T's, or they could be heterozygous. This one's purple and tall. Purple, tall, purple, tall, tall. This one here is purple, but it's short. This one's purple and tall. This one also is purple and short. This one's purple and tall, purple and tall. This one is tall, but not purple. Same with this one, tall, but not purple. Purple, tall. This one is not tall. This one is not purple, and this one is not purple. So you can see that I've crossed out all of the ones that are purple and tall. So now I can count them. 1, 2, 3, 4, 5, 6, 7, 8, 9. There should be 9 that have dominant phenotypes. Now let's count all of the ones that are purple but short. So here we've got purple and short, purple and short. This one's white and tall. This one's white and tall. This one is purple and short. Notice that I am not counting any of the ones that I already crossed off. Once it has been accounted for, you do not count for that individual twice. The rest of these are going to be white. So there are 3 purple and short. Now let's find all of the ones that are white but tall. So we've got little p is white, and then all it needs is 1 capital T. So this one is white and tall, white and tall, white and tall. This one's going to be white and short. So we're going to end up with 3 white and tall, and then this last one is white and short. So the typical phenotype ratio is 9 to 3 to 3 to 1. So every time you see a Mendelian dihybrid cross, you should expect 9 that are both dominant. These are dominant. 3 that are dominant and recessive, 3 that are recessive and dominant, and then 1 that is both recessives.